In a sealed Italian cathedral, a piece of linen cloth has been guarded for centuries by a secrecy so absolute that for decades, scientists were not even permitted to touch it. When they were finally allowed close enough to extract microscopic traces of genetic material from its fibers, what they found did not settle the argument that has divided historians, theologians, and forensic scientists for 100 years. It deepened it, because the DNA pulled from the Shroud of Turin did not point to one place, one person, or one simple explanation. It pointed to the entire ancient world at once. And the story of how that genetic evidence was found, what it actually says, and why it still cannot answer the one question everyone wants answered, is one of the strangest convergences of science, faith, and unresolved mystery in modern forensic history. The Shroud of Turin is a herringbone twill linen cloth measuring roughly 14 feet by 3.5 feet, bearing the faint, straw-colored image of a man, front and back, as though he had been laid on one half of the cloth, which was then folded over his body. The image shows wounds consistent with scourging across the back and shoulders, puncture marks around the head, consistent with a crown of thorns, and wounds at the wrists and feet consistent with crucifixion. For believers, this is the burial cloth of Jesus Christ. For skeptics, it is one of the most sophisticated forgeries, or one of the most persistent unexplained artifacts in human history. The Catholic Church itself has never officially declared the shroud authentic, treating it instead as an object of devotion whose scientific status remains formally unresolved. The modern scientific era of shroud research effectively began in 1978, when a team of American scientists working under the name STURP, the Shroud of Turin Research Project, was given unprecedented access to examine the cloth directly. Over 5 days of continuous around-the-clock analysis, the team used X-ray fluorescence, infrared spectroscopy, microscopy, and photographic analysis to study the image and the cloth's physical properties. They also used adhesive tape to lift microscopic particles, pollen grains, fibers, and trace organic material from the surface of the cloth for later analysis. Those samples, collected in 1978 and supplemented again during sampling in 1988, became the foundation for genetic studies that would not become technologically possible for another 3 decades. Before DNA testing entered the picture, the shroud had already been at the center of one of the most contentious scientific controversies of the 20th century. In 1988, 3 independent laboratories at Oxford, Arizona, and Zurich were given small samples cut from a corner of the cloth to perform radiocarbon dating. The results, published that year, dated the linen to a range of approximately 1260 to 1390 AD, placing its origin firmly in the medieval period rather than the 1st century. For many researchers, this seemed to settle the matter. The shroud, according to the dating, could not have wrapped the body of a man who died around 30 AD. But the controversy did not end there. Critics of the 1988 testing pointed out that the sample used came from a single corner of the cloth, an area some researchers argued may have been repaired or rewoven during the medieval period after fire damage the shroud sustained in 1532, potentially contaminating the sample with newer fibers. The debate over whether the radiocarbon sample was representative of the whole cloth has never been fully resolved to the satisfaction of everyone involved, and it remains one of the most disputed scientific results in the entire shroud controversy. It was against this backdrop of unresolved tension that genetic science entered the story. In 2015, Gianni Barcaccia, a professor of genetics and genomics at the University of Padua in Italy, led a team that approached the question from a completely different angle than radiocarbon dating. Rather than trying to date the cloth itself, they wanted to know who, biologically, had been in contact with it. Working from the dust particles, pollen grains, cell debris, and blood-like clots vacuum collected during the 1978 and 1988 sampling sessions, the team extracted and sequenced both plant chloroplast DNA and human mitochondrial DNA from across multiple sections of the cloth, including areas corresponding to the face, hands, and feet of the image, as well as the lateral edge used in the original radiocarbon testing. What came back from the sequencing was extraordinary in its diversity. The analysis detected human mitochondrial sequences from multiple individuals of different ethnic origins, clustering into haplogroups, genetic lineages traceable to specific ancestral populations that included groups typical of Western Europe, the Near East, the Arabian Peninsula, and the Indian subcontinent. This was not the genetic signature of one person. It was the accumulated biological residue of many different people, from many different parts of the ancient and medieval world, all of whom had at some point come into direct physical contact with this single piece of cloth. The breakdown the team published showed specific haplogroups including H1 and H3, common across Western Europe, alongside H13 and H33, typical of the Near East, R0A, associated with the Arabian Peninsula, and M56 and R8, linked to the Indian subcontinent. A later, expanded analysis pushed the numbers further. More than 55% of the detectable human DNA traced back to populations from the Near East, while approximately 39% corresponded to individuals from the Indian subcontinent, with smaller percentages linked to Europe and other regions. A broad cross-section of the ancient trade and pilgrimage world layered onto a single piece of fabric. The plant DNA told an equally sprawling story. Researchers identified plant species native to the Mediterranean, alongside species whose primary origin lay in Asia, the Middle East, and even the Americas, the latter only introduced to the wider world after the period of European contact with the New World. A cloth carrying the genetic fingerprints of plants from 4 different continents deposited across an unknown span of centuries is not the profile of an object that sat quietly in one place. It is the profile of an object that moved extensively, repeatedly, across an enormous geographic range, picking up biological hitchhikers from every region it passed through or every pilgrim who touched it along the way. This is where the genetic evidence becomes genuinely fascinating rather than simply confusing. Because the geographic spread of the human DNA, weighted so heavily toward the Near East and the Indian subcontinent, is not random. It maps remarkably well onto one specific historical theory about the Shroud's journey, a theory that predates the DNA findings by decades. Some Shroud historians have long argued that the cloth, if genuine, would have traveled from Jerusalem through Edessa in what is now southeastern Turkey, then onward through Constantinople, before eventually arriving in France and finally Italy in the medieval period, a journey of roughly 2,000 years across the heart of the ancient trade and pilgrimage routes connecting the Mediterranean, the Near East, and South Asia. The researchers themselves noted that while the genetic diversity does not exclude a medieval European origin for the cloth, it is also entirely compatible with the historic path the Shroud is traditionally believed to have followed during its purported 2,000-year journey from the Near East. But the most genuinely surprising thread running through both the original 2015 study and a more recent reanalysis involves India specifically. The proportion of human DNA traced to Indian lineages was high enough and consistent enough across multiple rounds of testing that researchers began asking a question nobody had seriously entertained before. Not just who touched the cloth, but where the cloth itself, the actual linen, might have been woven. The findings raised the possibility that the Shroud was manufactured in India rather than the Near East or Europe, a theory that initially sounds implausible until you account for the ancient textile trade. Researchers have pointed out that the word shroud derives from the Greek term sindon, meaning fine linen, a word some scholars argue may be linguistically connected to Sind, a historic region renowned for producing high-quality textiles, with well-documented commercial trade routes linking India and the Mediterranean world, stretching back into antiquity. The most recent phase of this research, building on material provided by forensic medicine professor Pier Luigi Baima Bologna, who had separately claimed decades earlier to have identified human blood of the AB type on the cloth, pushed the genetic analysis even further. The newer investigation confirmed the presence of haplogroup H33, a lineage prevalent in the Near East and notably frequent among the Druze, a population that shares common genetic ancestry with Jews and Cypriots, and has historically intermixed with other Levantine populations, including Palestinians and Syrians. The persistence of this specific genetic signature across multiple independent rounds of testing reinforced the broader hypothesis that the cloth had been exposed to numerous different populations over an extended period, rather than being confined to a single geographic origin at any one point in its history. It would be easy, at this point, to assume the DNA evidence simply confirms the Shroud's authenticity, tracing a clean line from 1st century Jerusalem to modern Turin. The actual scientists involved are far more careful than that, and their caution is itself one of the most important parts of this story. Barkacha himself, summarizing the findings directly, stated that individuals from different ethnic groups and geographical locations had clearly come into contact with the Shroud, whether in Europe, in Northeast Africa, in the Caucasus, in Anatolia, in the Middle East, or in India, but that the evidence could not say anything more definitive about the cloth's true origin. The findings, in his own assessment, do not rule out either the possibility that the linen is a medieval forgery or the possibility that it is the authentic burial shroud, leaving the central question of the shroud's true nature exactly as unresolved as it was before the genetic testing began. The most recent and most exhaustive reanalysis of the genetic material, examining samples collected from 12 different locations across the cloth, alongside threads originally used in the 1988 radiocarbon study, only deepened that uncertainty rather than resolving it. The team found an extraordinarily diverse array of medieval and modern DNA belonging to humans, animals, and plants, so varied that the field of metagenomics, the study of genetic material, recovered directly from an environment rather than from a single controlled source, could not provide definitive evidence regarding either the cloth's true age or its genetic origin. To test how contamination might occur and accumulate over time, the researchers even created an artificially contaminated linen sample for comparison, ultimately tracing the recovered plant DNA back to at least 19 distinct species common to the Mediterranean, alongside others native to China, the Middle East, and the Americas, while the human DNA pointed to traces from at least 14 separate individuals of differing geographic ancestry. What this body of research collectively demonstrates is something more unsettling, in its own quiet scientific way, than any single dramatic revelation could be. The Shroud of Turin is not a sealed, sterile artifact frozen at the moment of its creation. It is a porous, fiber-based object that has spent centuries, possibly 2,000 years, in direct contact with human hands, human breath, human tears, the open air of multiple continents, and the biological residue of everyone and everything that has ever touched, kissed, carried, or knelt before it. Popes have touched it. Pilgrims have touched it. Restorers, scientists, clergy, and curious hands across centuries of veneration and study have all left something of themselves behind, microscopic and invisible, but chemically permanent enough for modern sequencing technology to recover and read centuries later. This is precisely why the genetic evidence cannot deliver the single, clean answer that popular retellings of this story often promise. DNA does not carry a date stamp. A genetic sequence from a Near Eastern haplogroup found on the cloth could have been deposited by a first-century mourner in Jerusalem, or by a medieval crusader carrying the cloth home from Constantinople, or by a 20th-century priest from Lebanon attending a public veneration ceremony in Turin. The biological trace is real. Its chronological placement is not something genetics alone can determine. The same is true of the radiocarbon dating controversy from 1988, the blood type claims from the 1980s, the pollen analysis from decades of microscopy, and now the DNA sequencing from 2015 and beyond. Each method has produced real, peer-reviewed, scientifically rigorous data. None of them, individually or combined, has produced consensus. What the DNA actually revealed in the end is not a hidden code, not a secret identity, not a definitive answer to the question that has consumed Shroud research for over a century. What it revealed is a map of human contact spanning continents and possibly millennia, a genetic record of devotion, curiosity, and reverence layered onto fabric one invisible trace at a time. It revealed that whatever this cloth is, wherever it was made, and whoever or whatever its image actually depicts, it has been touched by an extraordinary cross-section of humanity across an extraordinary span of time and geography. The Near East, India, Europe, North Africa, the Caucasus, China, the Americas, all of it, somehow, present in the dust of a single 14-foot length of linen. The scientists who have spent decades on this research are not chasing a final verdict anymore, not in the way the public often imagines. They are documenting layer after layer of evidence, each one real, each one rigorously tested, each one adding detail without delivering resolution. The radiocarbon date says medieval. The blood analysis suggests something else. The pollen record points toward an ancient Near Eastern journey. The DNA points toward Near Eastern and Indian populations in numbers too significant to dismiss as coincidence. None of these threads fully agree with each other, and none of them have been definitively disproven. The Shroud of Turin remains exactly what it has been for as long as serious scientific inquiry has been pointed at it. Not a solved mystery, not a confirmed relic, not a debunked forgery, a genuinely contested object, sitting at the exact intersection of faith and forensic science where every new technique we develop adds more data and somehow, every single time, leaves the central question more open than it found it.